स्क्रीन विजिबल है आप लोगों को?
Yes, ma'am.
Okay. So let's start today's class. The topic is protein synthesis, and we have already talked about the initial part of the unit, that is unit 5, and we have already talked about translation machinery, ribosome, composition and assembly, universal genetic code, degeneracy of codons, termination codons, Google hypothesis. Now we will start the mechanism of protein synthesis. So we have already talked about the different steps of protein biosynthesis, and protein biosynthesis, it occurs in 5 different steps. The first step is activation of amino acid. Second step is initiation of the messenger RNA. Third step is elongation. Fourth is termination and release of polypeptide. Fifth step is folding and post-translational modification or processing. And this is a table which actually describes the components required for the 5 major stages of protein synthesis in E. coli. So we first talk about the prokaryotic protein translation, and the model system is E. coli. So in first step, that is actually the activation of amino acid and charging of transfer RNA. It requires different types of 20 amino acids along with different 20 amino acid transfer RNA synthetases. These amino acid transfer RNA synthetases, these are the enzymes which is actually required for the activation of amino acids and transfer of the amino acid on particular transfer RNA. Okay. Apart from these 20 amino acids and amino acid transfer RNA synthetase, the 32 or more transfer RNAs are also required. And ATP molecule, which is required to provide energy for the reaction, and magnesium ion, which act as a cofactor. Then next step is initiation, which requires messenger RNA. Then in case of E. coli, it requires N-formyl methionine transfer RNA, initiation codon in messenger RNA, that is AUG, which is universal for both prokaryotic system as well as eukaryotic system. Then ribosome, which consists of small and large subunits, as we have already discussed about, and different initiation factors, that is IF1, IF2, IF3. The energy which is provided to this reaction, this is initiation reaction, it is actually provided by GTP molecules. Remember, the energy in the initiation process is provided by ATP molecule, not by GTP molecule, not by ATP molecule. And the magnesium ion again used as a cofactor. It is clear? So next जो आपका step है, that is elongation. It requires functional 70S ribosome in its initiation complex form. Amino acid transfer RNAs, different types of, they must be charged and specified by codons. Then different types of elongation factor. So there is three elongation factor in case of E. coli, and GTP as a energy currency, and magnesium ion as a cofactor. Then fourth step requires termination codon in messenger RNA and different three types of release factor. Then folding and post-translational processing, it requires various specific enzymes, cofactors, and other components for removal of initiating residues and signal sequences. And also for proteolytic processing, modification of terminal residues, and phosphorylation of the particular protein, or any type of covalent modification which include the addition of methyl carboxyl carbohydrate or different types of prosthetic groups. So these are essential components required for different five major steps of protein synthesis in E. coli. So let's start with the first step, that is activation of amino acid. Okay. So in the first step, actually this step is also categorized into two steps. First step, that is the activation of amino acid, and the second step is the formation of amino acid acyl transfer RNA. So this reaction is actually catalyzed by an enzyme, specific enzyme, amino acid specific enzyme, that is known as amino acyl transfer RNA synthetase. And the overall reaction, it is depicted as the particular type of amino acid plus transfer RNA plus ATP, and in the presence of the enzyme amino acyl transfer RNA synthetase, the formation of amino acyl transfer RNA takes place with the release of AMP and 2-pyrophosphase. Is it clear to all of you? Kindly answer.
Yes, ma'am.
Yes, ma'am.
So this activation and transfer of amino acid to the transfer RNA, it is catalyzed by amino acyl transfer RNA synthetase, and these amino acyl transfer RNA synthetase, they are highly specific for different 20 types of amino acids. Okay, so they are classified into two classes. The overall amino acyl transfer RNA synthetases, they divided into two classes, class 1 and class 2. And this depends upon their substantial differences in primary and tertiary structure as well as in reaction mechanism, which we will see later in this discussion. ठीक है, ये आगे हम देखेंगे कि क्यों ये क्लास 1 और क्लास 2 दो क्लास बनाए गए हैं अमाइनो एसाइल ट्रांसफर आरएनए के, तो आरएनए सिंथेटेज एंजाइम के। तो ये एंजाइम अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेज, इट इज हाईली स्पेसिफिक फॉर ए पर्टिकुलर अमाइनो एसिड। ये ध्यान रखिएगा कि हर अमाइनो एसिड का अपना अलग अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेज होता है, which recognize the particular amino acid and its corresponding transfer RNA. Okay, and these two classes are same for all type of organisms. Is it clear to all of you? कहीं कोई दिक्कत हो अभी तक तो बताइएगा। This actually overall the protein synthesis, it is taking place in the cytosol, and amino acyl transfer RNA synthetases, they sterify the 20 amino acid to their corresponding transfer RNA. And each enzyme is specific, as I have told you, each enzyme is specific for one amino acid and one or more corresponding transfer RNA. ओके, हर एक अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेज which catalyze the esterification of 20 amino acid with their corresponding transfer RNA, ये स्पेसिफिक होता है फॉर वन अमाइनो एसिड एंड फॉर वन और मोर करस्पॉन्डिंग ट्रांसफर आरएनएज। इसीलिए 20 अमाइनो एसिड के लिए 20 अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेजेस हैं और 32 और
मैम
मोर
वॉइस
देन।
क्लियर नहीं आ रहा।
बेटा ये आपके साथ भी प्रॉब्लम हो सकती है क्योंकि मैंने 2 सिस्टम ऑन कर रखे हैं और मुझे वॉइस क्लियर आ रही है। सबके साथ है ये प्रॉब्लम?
No, ma'am.
नो मैम।
No.
So kindly check out your check your internet, ठीक है, कि कैसे ये आ रही है आपकी इंटरनेट सर्विस। सो there are 20 amino acyl transfer RNA synthetase specific for 20 amino acid, and they recognize more than 21 amino acyl transfer RNA synthetase. It recognize one or more than one type of transfer RNA, करस्पॉन्डिंग ट्रांसफर आरएनए। ठीक है, इसीलिए जो ट्रांसफर आरएनए की जो टोटल नंबर मैंने दिखाया है शुरू में, वो आपके मोर देन 32 है। ठीक है, जबकि अमाइनो एसिड आपकी 20 है। So most organisms they have one amino acyl transfer RNA synthetase for each amino acid, and for amino acids with 2 or more corresponding transfer RNA, the same enzyme, it is usually amino acylate all of them. So as I have told you that these amino acyl transfer RNA are further categorized into 2 classes based on their differences in primary and tertiary structure as well as in reaction mechanism. And these 2 classes are same in for all organisms, that is ranging from bacteria to mammal system. And the reaction catalyzed by amino acyl transfer RNA synthetase, it is given over here. That is amino acid, it reacts with transfer RNA in the presence of enzyme and ATP molecule and form amino acyl transfer RNA. And this reaction occurs in 2 steps in the enzyme's active center. So here is the detail of the reaction. This is the amino acid. ये ATP molecule है. So the first step in this reaction after binding of this amino acid to the enzyme active site. Enzyme का जो active site होता है, जिसमें 2 साइट प्रेजेंट होते हैं, एक बाइंडिंग साइट एंड कैटालिटिक साइट। तो जो अमाइनो एसिड स्पेसिफिक बाइंडिंग साइट है, वहां पे स्पेसिफिक अमाइनो एसिड जाके बाइंड करता है। एंड द फर्स्ट स्टेप इज फॉर्मेशन ऑफ अमाइनो एसाइल एडनाइलेट या अमाइनो एसाइल एएमपी। This amino acyl AMP, it is a reaction intermediate, okay, and it is also known as enzyme bound intermediate, and it forms when this कार्बोक्सिल ग्रुप ऑफ अमाइनो एसिड, कार्बोक्सिल 2 एंड होते हैं, अमाइनो एंड कार्बोक्सिल एंड द कार्बोक्सिल ग्रुप ऑफ द अमाइनो एसिड रिएक्ट्स विद द अल्फा फास्फोरिल ग्रुप ऑफ द एटीपी। दिस इज द एटीपी, ये एडनिन है, ये राइबोस शुगर है, एंड दिस इज द 3 फॉस्फेट ग्रुप अल्फा, बीटा and gamma. So this carboxyl group, it attacks on the alpha phosphoryl group of ATP and form an anhydride linkage with displacement of pyrophosphate. ठीक है, PPI, that is known as pyrophosphate. So formation of 5 prime amino acyl adenylate takes place. formation of amino acyl 5 prime amino acyl adenylate. It is takes place. This is the first step of amino acylation of transfer RNA by amino acyl transfer RNA synthesis. Is it clear? समझ में आ रहा है?
Yes, ma'am.
In the second step, which depends upon the class of the amino acyl transfer RNA synthetase कि class 1 है या class 2 है. ठीक है. So the attachment of amino acyl to the corresponding specific transfer RNA takes place. So in the second step, the amino acyl group from this amino acyl adenylate, it is transferred from to its corresponding specific transfer RNA. And this second step, it depends on the class to which the enzyme belongs. So here, different mechanism for different class has been depicted. So first we will take the class 1 amino acyl transfer RNA synthetase enzyme mechanism. In this process, you can see here the amino acyl जो AMP होता है, that is enzyme bound adenylate amino acyl adenylate, it is attacked by 2' hydroxyl group of the ribose sugar. This जो CAA amino acid binding ब्रांच होती है ट्रांसफर आरएनए की, उसका जो टर्मिनल न्यूक्लियोटाइड होता है दैट इज CAA. उस CAA के जो फाइनल एडनाइलेट होता है, एडनाइलेट का 2' हाइड्रोक्सिल ग्रुप, इट अटैक्स ऑन द carbon of the carboxyl group of amino acid in amino acyl AMP complex. So there is the bond shifting which results in the formation of amino acyl transfer RNA, but here the amino acyl first it binds on the 2' position of the ribose sugar, and that is then after transesterification reaction it is shifted to the 3'. Okay. So the amino acyl group is transferred initially to the 2' hydroxyl group of the 3' terminal A residue of transfer RNA, then to the 3' hydroxyl group by a transesterification reaction. Is it clear to all of you? समझ में आया कि फिर से समझाना पड़ेगा?
एक बार फिर से बता दीजिए थोड़ा सा, ये लास्ट का.
दो स्टेप में जो पहला आपका अमाइनो एसाइलेशन ऑफ ट्रांसफर आरएनए जो फर्स्ट स्टेप है प्रोटीन सिंथेसिस में, दैट इज कैटलाइज्ड बाय अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेजेस. ये फर्दर 2 स्टेप प्रोसेस है ये रिएक्शन. पहले स्टेप में एक्टिवेशन ऑफ अमाइनो एसिड टेक्स प्लेस इन विच अमाइनो एसिड it forms an enzyme bound 5' amino acyl adenylate, that is also known as amino acyl AMP. This is known as activation of amino acid. This active amino acid then recognized by the specific transfer RNA by and bound with the specific transfer RNA by esterification reaction, in which initially in case of class 1 amino acyl transfer RNA synthetase, that 2 prime hydroxyl group, it actually attacks on the carbon of carboxyl group of amino acyl AMP, and the amino acyl group is transferred initially to the 2 prime hydroxyl group. 2 prime hydroxyl group of 3 prime terminal एडेनिन रेसिड्यू, जो सी ए आपका यहाँ पे 3 प्राइम ब्रांच दिखी थी, आर्म जो सी ए आर्म थी, उसके टर्मिनल अमाइनो एडेनिन रेसिड्यू को ये अमाइनो एसाइल ट्रांसफर हुआ एट द पोजीशन ऑफ 2 प्राइम हाइड्रोक्सिल ग्रुप इनिशियल, देन to that 3 prime hydroxyl group of the same adenylate group by a trans-esterification reaction. And finally, the amino acyl transfer RNA has been formed. Okay, this amino acyl transfer RNA is formed. And in class 1, it is a 2-step process, while for class 2, amino acyl transfer RNA synthetase, it is a single step process where amino acyl group from amino acyl AMP is directly transferred to the 3 prime hydroxyl group of terminal adenine of this transfer RNA, and formation of amino acyl transfer RNA takes place with the release of AMP in both cases. जब अमाइनो एसाइल AMP से अमाइनो एसाइल का ट्रांसफर होगा ट्रांसफर RNA पे, देन AMP it get release. समझ में आ गया?
यस मैम. मैम इसी को हम चार्जिंग ऑफ टीआरएनए बोलते हैं क्या?
हाँ, this is known as charging of transfer RNA. Okay?
Okay मैम.
Okay, ma'am.
So the pyrophosphate जो ये चार्जिंग होती है, चार्जिंग ऑफ ट्रांसफर आरएनए विद अमाइनो एसिड, इट इज ए हाईली नेगेटिव reaction. It means that it has a highly negative free energy of hydrolysis, and this amino acyl transfer RNA synthetase, it is very specific, and it shows the proofreading. Proofreading means if an incorrect transfer RNA or amino acid, it is bound with the enzyme, then enzyme should possess some mechanism to recognize the incorrect bound amino acid, so that it remove the incorrect incorporation of, or it to so that it avoid the incorrect incorporation of amino acid in a particular protein synthesis. ठीक है, तो अमाइनो एसाइल ट्रांसफर आरएनए सिंथेटेजेस, they possess proofreading also. Here in this slide you can see the detailed structure of amino acyl transfer RNA, in which an amino acid, it is bound to the 3 prime hydroxyl group of the terminal adenylate of CAA arm of this transfer RNA, and proofreading of this amino acyl transfer RNA synthetases, it has this amino acyl transfer RNA synthetases, it possesses proofreading, and it check the incorporation of correct amino acid in the protein by different 3 types of filter. And the first filter is the initial binding of the amino acid to the enzyme and its activation. because each and every amino acid transfer RNA synthetase, it recognize only a specific type of amino acid. So the first is the to recognize the specific amino acid and binding of this amino acid to the specific binding site of enzyme and its activation to amino acid AMP. अगर इनकरेक्ट बाइंडिंग हो गई फॉर इनकरेक्ट अमाइनो एसिड एएमपी प्रोडक्ट to a separate active site on the enzyme, so the substrate, that is amino acids, it is hydrolyzed by the enzyme itself. A substrate that binds in this second active site is hydrolyzed. So each transfer RNA synthetase, amino acid transfer RNA synthetase is highly specific for particular amino acid which it recognize and bind. For example, if we talk about the valine and isoleucine, तो वैलिन और आइसोल्यूसिन में बहुत ही माइनर डिफरेंस होता है. वैलिन आइसोल्यूसिन में एक मिथाइल ग्रुप एक्स्ट्रा होता है. ठीक है, तो वैलिन और आइसोल्यूसिन दे डिफर बाय ओनली ए सिंगल मिथाइलिन ग्रुप. However, the isoleucine transfer RNA synthetase, it recognize the isoleucine only. So it favors the activation of isoleucine over valine, and this favor is by a factor of 200. Okay, and there is a chance that valine may occupy a position in a protein where isolation should be present. So the frequency of this incorrect incorporation of valine in the place of isoleucine is only 1 in 3000. So this accuracy is very high, and it shows that amino acid transfer RNA synthetases are highly specific and has a proofreading function. So in the third filter, that is you can say the third point where the this enzyme can recognize the incorrectly incorporated amino acid. It is that most of the amino acid transfer RNA synthetases, they hydrolyze the ester linkage between amino acid and transfer RNA in the incorrectly charged transfer RNAs. तो अगर इन दोनों फिल्टर से बच गया तो यहाँ पे जब अमाइनो एसिड से ट्रांसफर आरएनए चार्ज होता है और अगर गलत अमाइनो एसिड आ गया, then that ester bond is hydrolyzed by the enzyme. So it provides a third filter to enhance the fidelity of the overall process. Is it clear to all of you? समझ में आ गया भाई? बताओ हाँ या ना. आप लोग प्रेजेंट हैं?
यस मैम.
Yes, ma'am.
समझ में आ
Yes, ma'am.
रहा है जितना बताया गया है कि कहीं कोई
Yes, ma'am.
यस मैम.
Yes, ma'am.
प्रॉब्लम है?
Yes, ma'am.
So the overall error rate of protein synthesis, it is 1 mistake per 10 to the power 4 amino acid. incorporated, and it is quite low. एंड अगर इसके बाद भी कोई प्रोटीन अब मतलब इनअप्रोप्रिएट सीक्वेंस जो है प्रोटीन के आते हैं या डिफेक्टिव प्रोटीन आती है, देन दैट प्रोटीन is degraded and not passed on to the future generation. There are many mechanisms which recognize the defective proteins, and they are actually specific or specialized for degradation of defective protein. So the degree of fidelity in protein synthesis is sufficient to ensure that most protein contains no mistakes. So this is the first step of the protein synthesis. The next step is initiation. So specific amino acid initiate protein synthesis. As I have told you that the initiation codon is the universal AUG, and in case of prokaryotic system, uh, N-formyl methionine is responsible for the initiation of the protein synthesis, while in case of this eukaryotic system, the methionine is responsible for initiating the protein synthesis. So, protein synthesis, it begins at the amino terminal end and proceed by the stepwise addition of amino acid to the carboxyl terminal end of the growing polypeptide. And this finding or this study, the credit for this study goes to Howard Dentizes, who had studied this in 1961. And here you can see in the slide that 30th subunit, it possesses P site and A site. P site is specific for what?
स्टार्ट कोडन मैम, इनिशिएशन है स्टार्ट कोडन।
इनिशिएशन एंड क्या कहते हैं पी साइट को?
ये मैम इस पे पेप्टाइडिल साइड बोलते हैं, पेप्टाइडिल साइड।
कहाँ?
ये तो पेप्टाइडिल साइड बोलते हैं मैम इसे शायद।
पिछली बार बताया था ना राइबोसोम का स्ट्रक्चर?
यस मैम।
क्या बताया था बताइए, थोड़ा सा बताइए फिर आगे बढ़े। This is the initiation formation of, this slide shows the formation of initiation complex in E. coli. This is the peptidal site. This is known as amino acyl site. And the first step is in the formation of this initiation complex. The first step is the binding of initiation factor 1, initiation factor 3 to the 30S ribosome subunit, followed by binding of messenger RNA in such a way that the initiation codons it comes at the peptidyl site. ठीक है. This step, the complex formation is, it takes in 3 steps, and the energy is provided by GTP, and GTP it is hydrolyzed into GDP plus PI, and these three initiation factor IF1, IF3, and IF2, they actually responsible for formation of initiation uh, complex, and they are known as initiation factor. So IF1 it first bound with the amino acid site, then messenger RNA. So in the first step, the 30S ribosomal subunit binds to initiation factor IF1 and IF3, and factor IF3 it prevents the uh, 30S and 50S subunits from combining prematurely. The messenger RNA then binds to the 30S subunit, and the initiating 5' AUG, it is guided to the correct position by a sequence known as Shine-Dalgarno sequence. Okay, which is named after an Australian scientist, Shine-Dalgarno sequence. These two researchers, they have actually studied for the initiation of this protein synthesis by binding of this AUG on the correct position, that is peptidyl position. And the consensus sequence is an initiation signal of 4 to 9 purine residue, and the sequence base pairs with a complementary pyrimidine rich sequence near the 3' end of the 16S ribosomal RNA of 30S ribosomal subunit. This messenger RNA and ribosomal RNA interaction, it is actually responsible for positioning the initiating codon of the messenger RNA in the precise position on the 30S subunit where it is required for initiation of translation. So the transfer RNA, which is responsible for recognizing this initiation codon, is N-formyl methionine transfer RNA, and it is to be bound, is distinguished from other methionine codons by its proximity to the Shine-Dalgarno sequence in the messenger RNA. So, as we know that this bacterial ribosome, it possesses 3 sites that bind amino acid transfer RNA, that is known as A site, the peptidyl site, and then exit site. Okay. So समझ में आ रहा है भाई? आंसर करिए. इतना समझ में आ गया?
Yes, ma'am.
फर्स्ट स्टेप प्रोटीन सिंथेसिस का क्या था? एक्टिवेशन ऑफ अमाइनो एसिड एंड चार्जिंग ऑफ ट्रांसफर आरएनए. ठीक है, तो पर्टिकुलरली इट इज द
यस मैम.
चार्जिंग ऑफ ट्रांसफर आरएनए विद द करेक्ट अमाइनो एसिड. सेकेंड स्टेप क्या है? फॉर्मेशन ऑफ इनिशिएशन कॉम्प्लेक्स. This formation of initiation complex, it is started with binding of two initiation factor onto the 30s subunit, that is IF1 and IF3. IF3, it is responsible for inhibiting the immature complex formation, that is the binding of 50s and 30s ribosome subunit together uh, uh, before the binding of the messenger RNA and recognition by transfer RNA. ठीक है, तो premature binding of 50s subunit to the 30s subunit, it is prevented by the presence of initiation factor 3. ये समझ में आ गया, हाँ या ना?
हाँ मैम.
ठीक है, then a sequence which is present in the messenger RNA, and that sequence is recognized by the two scientists, and hence the name Shine-Dalgarno sequence. And this Shine-Dalgarno sequence is, it is an initiation signal of 4 to 9 purine residues. And it is 8 to 13 base pair to the 5 prime side of the initiation codon. And this sequence, ये जो सीक्वेंस चाइल्ड डायग्राम सीक्वेंस होता है, जो कि एक तरह से सिग्नलिंग सीक्वेंस होता है, इट रिकॉग्नाइज एंड बेस पेयर with a complementary pyrimidine rich sequence near the 3 prime end of the 16s ribosomal RNA, or 16s ribosomal RNA, it is a component of 30s ribosomal subunit. ठीक है, द फॉर्मेशन ऑफ मैसेंजर आरएनए एंड राइबोसोमल आरएनए, क्या कहते हैं, हाइब्रिड, इट एक्चुअली, और द मैसेंजर आरएनए राइबोसोमल आरएनए इंटरेक्शन, it actually
हमारी ऑनलाइन क्लास चल रही है
positions
अभी
the initiating AUG sequence of the messenger RNA in the
गूगल क्लास रूम
precise, or you can say correct position, and the position is the P side.
पूरा रिचार्ज करवाया था सुबह खत्म हो गई उसमें बॉल ये
हाँ, क्या हुआ बेटा? टाइम ओवर हो गया क्या क्लास का? अमन, आर यू देयर?
यस मैम.
क्लास ओवर
मैम उसका
करें,
माइक ऑन हो
खत्म
गया था.
करें, है? चलिए बंद कर रहे हैं क्लास. नेक्स्ट क्लास आपकी फिर आपकी फ्राइडे को होगी. ठीक है,
ओके मैम.
Okay, ma'am.
Okay, ma'am.
Okay, ma'am.
ओके.
Thank you, ma'am.
अटेंडेंस अपनी
Thank you, ma'am.
गूगल क्लासरूम पे डाल दीजिएगा, और इसका जो है स्क्रीनशॉट लेके जो अटेंडेंस है ये भेज दीजिएगा मुझे. ठीक है.
Okay, ma'am.
हम्म.
Thank you, ma'am.
Thank you, ma'am.